Okay, I'm going to call to the front the next presenter, which is Mama Kuku. I hope I'm pronouncing things right.
Good afternoon, everyone. I'm Mr. Mama Koko MS. I'll be presenting to you my study on the incidence of stillbirth across generation in the South African Holstein dairy cattle. I've done the study under the supervision of Prof. Tada and Prof. Diaz. This is the outline of my presentation. It starts from the background going all along to the acknowledgements. I'll start by the background. South African industry comprises of various economic activities with significant difference in farming method and procedure of the dairy production. And it also production and the marketing of raw milk, pasteurized milk, and cream. In all the dairy breeds, one of the most economical and ethical important traits is the stillbirth. And among all of the South African Holstein dairy industry, the dairy cattle breeds that is most dominant is the Holstein dairy cattle, while there are some of those that are being used, which includes the Jersey, the Ayrshire, and the Guernsey breed. However, South African dairy cattle milk production contribute approximately 0.5% to the world milk production. And it also contributes 13% of the 2.3% contribution of the agricultural sector towards the GDP. The problem statement, incidence of stillbirth increases with about 6% to 11% during the past 25 years in the Holstein breed. And that indicates the problem associated with enhancing milk production, as the increase in the incidence of stillbirth normally result in some reduction in the milk production. Stillbirth increases is associated with increasing production cost due to premature culling, veterinary services, dam and calf mortalities. And ultimately that will result in undesired reproduction and the production of the enterprise. That will also include what deterioration in milk yield, longevity of the cow, increased cost of replacement stock. Globally, in the population of dairy cattle, the increase of the stillbirth age is one of the major problems. The rationale, incidence of stillbirth in the Holstein dairy cattle is very detrimental to the productivity of the enterprise and the milk industry at large. Stillbirth is classified as a trait of economic importance, as it knows that the knowledge of the cost and the economic losses that are related to it might assist in making better decision making and implementing those management practices which are proper that can reduce the incidences of stillbirth. That necessitates the need to evaluate the stillbirth incidences and the phenotypic trends across the generation of the South African dairy cattle. The aim of the study was to evaluate the incidences of stillbirth and the phenotypic trend across the generation of the South African Holstein dairy cattle. The objectives is that we're looking at determining the stillbirth incidences and the phenotypic trends across the generation of the South African Holstein dairy cattle. Taking you through the methodologies, the study side. The study was done in South Africa. That was including the data collected all over the South Africa as large from all the 9 provinces. It included all the animals recorded throughout the South Africa at large. And South Africa, it occupies the southern tip of Africa. The experimental animal was the Holstein dairy breed, which is the one that I indicated earlier, that is the dominating breed in the cattle dairy industry. And the Holstein breed of is known for its great milk production, and it weighs about 680 kilograms and stands about 140 centimeters at the width. Animal management. Animal in small-scale farming were managed conventionally. Animal husbandry practices including putting animals on a total mixed ration, which included 51% of forage and 49% of concentrate. The cattle were vaccinated against common diseases, which include the brucellosis, bovine viral diarrhea, infectious bovines and leprosis, and bovine respiratory syndrome virus. Animals were housed in holding that allow having shelter, allow easy handling, feeding, breeding, milking, and health activities. Replacement, heifers, selection, and breeding. That was based on the breeding values, on looking at the productivity potential. And pure breeding, using the good quality semen sourced at international market, was used during the breeding process. Study design. The study design that was used was the longitudinal observational study, as the study was done during a long period of time and it didn't have any particular treatments. Data collection. The study used the secondary data that was extracted at integers database, which included about 1 million Holstein dairy cattle born from 1945 to 2018. And in that, we did data pruning whereby we're looking at removing animals with incomplete records. Incomplete records, we look at those that didn't have birth weight in the records and other things that we're looking at. We also removed the herds that has less than 10 animals. That resulted to a total of about 380,157 cows from 1,767 heads. Information that were gathered over different generations included pedigree, data, sex cell, dam factors, and the birth status. Stillbirth was recorded as 1 for alive and 1 for death. The birth season and the birth year effect were derived from the dam calving dates. Statistical analysis, the data editing was done. That included the preliminary data editing and the advanced data editing. The preliminary data editing was done using Microsoft Excel, and while the advanced data editing was done using the Sage Vision 1.9. Average generation interval were computed using the formula indicated here, while the incidence of stillbirth across generations were modeled using fixed factors and the random factors. This is the model that was used at 5% significant level, while the phenotypic trends were determined from mean values based on the calculation generated. This is the model that was used. Taking you through the results and discussion, here we have the POX plot. This is the POX plot of the stillbirth percentages. This shows that there was more heads that had about 0 incidences of stillbirth, while some had about 100 incidences of stillbirth. That indicates that there was a variation or a very high variation. As you see that it actually ranges from 0 and to 100, while most of the heads, they have concentrated close to 0% of stillbirth. Here I'm going to continue to effect of dam parity on instance of stillbirth across generation, of course. Here it showed that from the first parity to the 12th parity, there is a slight decrease from the first parity going down until the 6th, 7th. However, when it starts to 9, it starts to increase, whereby it shows that actually at first calving, there is at the first calving compared to the 12th calving or parity, it shows that at first there is 4%, which is less than the 10% that was shown at the 11, 12 parities, which shows that those that are the young cows actually have lower incidences of stillbirth compared to the old ones. This is the effective population to season and the calf sex. It shows that during the season, it shows that the summer and the autumn has a higher significant effect, while the others didn't have, they had the lowest. The sex, the female had the highest, while the male had the lowest. This is the correlation table. It showed that from the 10 correlation, only 6 had an effect, while the other 4 were not significantly effect. Conclusion and recommendation, there was a great variation in the incidence of stillbirth that I've indicated that can be due to environmental condition, different head size, genetic variation, and management practices. Also that we saw that the young animals had the lowest, while the female also had the lowest association to incidence of stillbirth. That showed that can be due to the proportion of the pelvic area and the calf size. However, there is this high variation of incidence of stillbirth across the head in the small-scale farming that warrant a need for further investigation in the inbreeding rate and the effective size within the South African Holstein dairy cattle population. These are some of the references that are used in doing this study. I would like to give acknowledgement to the Limpopo University and the Department of Agricultural Economics and Animal Production, and together with all the groups and individuals I work with in conducting the study. That marks the end of my presentation. Thanks.
Thank you, Mr. Mamakuku, for a nice presentation. Any questions? I know people that you are coming from lunch, but I'm begging you this time. Any questions? Okay, going once, going twice. Mr. Mamakuku, I'll have to do my job as a chair and ask you a few, ne? Okay, yeah. So, oh, there is a question. Oh, okay, there's a question that side.
What would you put forward as your explanation for the increased incidence of stillbirth as parity increased?
I can hear. Can you speak to the mic? I can hear.
We saw the parity going up.
Yes.
Once parity was, we saw the stillbirth going up as parity reached like 10, 11.
Yes.
What would you offer as explanation for that observation?
For that?
Yeah.
Okay. Normally, when the cows, they normally have a certain age group whereby after a certain number of parities or a certain period of time, they start having some issues as they age. Mostly they have complications during paturation, meaning that some of the causes can be the challenges that are faced during paturation, health issues. Yeah, that's the contributing factors.
Are there any questions? Otherwise, I will allow follow-up questions this one time. Okay. In the absence of follow-up questions and any questions, let me ask you a question. You say that you used data from integers.
Yeah,
integers, sorry, my bad. And this is secondary data, you say. Yes. Are there any notable limitations to using this secondary data? That if you use primary data, maybe, things would be better? Are there any notable limitations to using the secondary data?
I think the challenge, the limitations may be due when the way the data was recorded, maybe, as some of the recordings were not really okay. Because some heads have like maybe 4 animals, or some had only 2. So if there are only 2, you cannot really yield a good or a significantly accurate conclusions from that, because you only have 2 animals, then maybe all of them, they have stillbirth. So you cannot really say you had 100% stillbirth instance within a head where you only had maybe 2 animals. So those can be some of the limitations that I can say they were there in conducting the study.
Always for you, anytime.
Yes, hence I'm saying those are some of the challenges that came into play with handling the data.
Thank you, Mr. Mamakuku. Yeah, I need this as well. Okay, can you please clap hands? Okay, before I call our next speaker, I just need to do a bit of admin. There are people I've been told who need to give posters soft copies. Some of them, it could be that they need to give us hard copies. I'm going to ask that you wait, and then during our next tea session, you come straight to yours truly, and you bring particularly your soft copies. But if you've got hard copies as well, I need to know from you whether they've been put up there or not, right? So if you're one of those people, you know yourself, please come see me afterwards. We're going to jump to our next presenter. And our next presenter is going to be Miss Masipa, if I'm correct. I think I've already gotten the titles wrong. There was someone who was wearing a ring. I called them a miss, so my apologies there. Miss Masipa, please come up. I see 2 fingers from you. It's all systems go.
Good morning, sir. Good afternoon, ladies and gentlemen. My name is Kharolo Masipa, and I'll be presenting to you the findings of my study titled Chicken Compost Amendment and Manipulation of Water Regimes Suppresses Fusarium Mold of Tomato and Influences Plant Growth Parameters. Soil-borne diseases have been managed by manipulating the biochemical properties of the soils. In recent years, we've seen that interest has quickened when the role of soil physical properties on disease suppression became apparent. This is because in addition to being a storehouse for the nutritional needs of microbial communities, the soil physical properties also provide the physical habitat in which the soil biological interactions take place. However, not all soils can naturally meet these requirements. As such, the incorporation of chicken compost as well as water into the soil will lead to soil improved biota, which will then improve the soil conditions that affect plant growth. Now, the prevailing soil conditions that affect plant growth can and will influence the severity of soil-borne diseases, including Fusarium mold of tomato. However, despite all that has been said, the effect of the changes in physical properties on disease suppression remains poorly understood. With that in mind, we sought out to answer the question, how does chicken compost amendment and manipulation of water regime affect soil properties, plant growth parameters, as well as the suppression of Fusarium mold of tomato? We aim to investigate the effects of amending soil with varying chicken compost and soil moisture regimes on the development on the Fusarium mold of tomato, with the objective to quantify the effect of amending soil with chicken compost and water regimes on physical properties, plant growth parameters, and the development of Fusarium mold. The current study was conducted at Green Biotechnologies Research Center, which is located at the University of Limpopo, and that shows the location map. In preparation for the greenhouse trial, soil was collected from the surface layer at a depth of 0 to 20 centimeter. The compost was collected from a broiler production house. The 2 materials were then taken to a soil mixture to be thoroughly mixed. The experiment was laid out in 3 chicken compost levels by 3 water deficit levels in a randomized complete block design. 4 weeks old tomato seedlings were then transplanted into the soil pots in soil pots that contain different mixed ratios of compost and water levels. 5 grams of Fusarium oxysporum rice grains was used to inoculate our soils. At the end of the experiment, soil physical properties that were determined included bark density, porosity, airfield porosity, waterfill porosity, and gravimetric soil moisture content. Measured plant growth parameters included plant height, stem diameter, plant biomass, as well as the leaf chlorophyll content. Shoot symptoms as well as stem and root discoloration was assessed on disease severity scales. All statistical analysis was performed using GenStat, wherein a two-way ANOVA was employed to compare the effect of chicken compost and water holding capacities on measured soil and plant parameters. Duncan multiple range test was further used to separate against significant treatment means. Ladies and gentlemen, the applied treatments yielded varying effects on measured parameters. There was a significant decrease in bug density with increasing rates of compost and water holding capacity. Now this indicates that the decrease in bug density was driven by higher application rates of compost. As expected, soil porosity increased with decreasing bug density. This is related to the pore size distribution of the soils. In this respect, we have seen that soil micropores and mesopores increased while the macropores decreased, and macropores are known to lead to higher porosity. To further understand, to understand the effect of porosity on our soils, we then looked at air-filled porosity as well as water-filled porosity. There was an increase in water-filled porosity with increasing rates of compost and water holding capacity. The high rates of air-filled porosity in compost-amended soils indicated that there was improvement in aeration. On the other hand, there was a decrease in water-filled porosity. The high values of water-filled porosity in no amended soils indicated that the airspace in no amended soils was taken up by water. There was no significant increase in the gravimetric soil moisture content as depicted in the figure, except for soils that were optimally treated. However, Palmer in 1995 argued that the differences in soil moisture content should be given priority over the statistical differences due to the effect that they have on water storage capacity. Ladies and gentlemen, our plant height, stem diameter, fresh shoot weight, fresh root weights, and the chlorophyll content of the leaves significantly increased with increasing rates of compost and water holding capacity. This was ascribed to the improved aeration, which allowed plants to access resources necessary for plant growth. As depicted by the red arrows, Fusarium mold of tomato was suppressed following increasing rates of compost and water holding capacity. What we know from the literature is that the ability of soil-borne diseases, including Fusarium mold, to manifest in soils is dependent on soil physical properties. What we observed in the current study is that there were reduction in bug density, which then improved the aeration and increasing the soil moisture content. This allowed plants to access resources through greater presence of roots, which were observed in plants that were amended with compost. This then led to the suppression of the disease. In conclusion, we observed that changes in soil abiotic factors played a key role in promoting plant growth parameters. This study further highlighted that when optimally applied, chicken compost and water holding capacity led to effective disease suppression. What still needs to be done from this study is that we still need to account for the microbial communities as well as the nutrients of the studied soil, because we don't want to isolate the physical property. We don't want to put physical properties in isolation. Ladies and gentlemen, allow me to acknowledge my supervisors, Prof. P.E. Dlamini, Prof. M.A. Kena, the National Research Fund, as well as the University of Limpopo. Thank you.
Thank you very much for that nice presentation. I'm going to make my plea again for questions.
Thank you very much for that interesting presentation. I'm not sure if I missed it or not. With the soil that you used, did you sterilize it prior to use in the experiment?
Oh yes, you didn't miss it. Sorry, you didn't miss it. I didn't include it in my slides, but we did pasteurize the soils.
Okay, super.
Okay, this was a factorial experiment, right?
Yes, sir.
Did you investigate the existence or lack thereof an interaction between your factors?
No, we didn't do any interaction.
Do you think that's the right way to look at it?
Sorry?
I said, do you think if it's a factorial experiment, what would you, or how do you feel about it? Would it be better for you to look at whether your two factors are interacting? Because that's the whole reason of doing an interaction study.
Yes.
Because maybe when you restrict water and you are increasing your compost, it might have a negative effect on your plant.
Yes.
Whereas when you increase both, you might have a positive feedback. So you might want to look at that interaction just to see what it looks like. It might change how you interpret your results.
Yes. Thank you.
Just a quick one, Chairperson. You use the factorial with the block design.
Yes.
What was the reason for blocking?
The experiment was conducted in a greenhouse where we had wet walls on the other end, and there were we were also blocking for the wind currents that were blown by the thermostatically controlled fence.
Okay, so this wind was not affecting the whole plot.
Can you please repeat?
The wind was not affecting the whole plot.
I would like to believe that if we were to move our experiments towards the wind, it wouldn't give us the same results as we were to move it to the other end. So that's why we had to block for it.
Okay, thanks, Chairperson.
Thank you. You were very generous with time. I think you took 7 minutes, 30 seconds. That's just to the adjudicators as well. So I'll have to throw in a question or two because you were generous. I was told I have to. Okay, so you say that you had 3 water regimes.
Yes.
How did you get your water holding capacity or your field capacity? How did you measure that?
Oh yes, I didn't include it in my slide, but the determination of water holding capacities was done on a mass basis, wherein we took 3 replicates of the soil pots. We filled it with 3 grams of water. We then used a measuring cup to keep track of the amount of water it takes to saturate the soil. So we then we let the water to drain for 24 hours and weighed the mass of the pot. This was again confirmed for after 48 hours.
So basically what you were measuring was field capacity.
Yes, it was soil capacity.
Okay, I'm not a soil scientist, but from my experience,
Yeah,
there are more accurate measures of doing that.
Yes.
Yeah, but maybe we don't have the equipment
No,
at the university.
yes.
My last question, given that you gave us so much time, is you say that there's a relationship between soil-borne diseases. You say it earlier, and you also say it in your results. There's a relationship between soil-borne diseases, and that relationship extends to the soil conditions,
Yes.
right? What is that relationship? You never truly expanded. How is it that if you improve soil properties, you suppress diseases? It was never clear in my view.
Can you please repeat?
Okay,
There's a
so
relationship between
your results say that if you improve, if you reduce bulk density and you improve porosity, you suppress the diseases, the fusarium specifically.
Yes.
How does that happen? If you
Okay,
can
in
expand.
my introduction, I think I was brought in that I said there's a relationship between the soil-borne disease and physical properties, because I didn't expound on which soil properties specifically. So when you look at the properties that I then looked at, you see that when we decrease the bulk density, there is an increase in porosity. So if I said there is a positive there is a positive relationship with the physical properties, in the sense that when we decrease bulk density, we suppress the disease.
Can you please clap hands? Yeah, I think you are the first person after lunch who's had so many questions thrown at you, so thank you for handling that. Thank you for handling that. We're going to jump straight to our next presenter, and our next presenter is going to be Ms. Manyike. Please come to the front and see Prof. Chasi. You see 2 fingers, all systems go.
Okay. Good morning, ladies and gentlemen. My name is Butar Manyike, and I will be presenting my research titled Heavy Metal Contamination of Soil at the Weltervreden Landfill Site, under the supervision of Professor P.E. Dlamini, Professor M. Mujuru, and Professor V.L. Mlaudzi. Municipal solid waste refers to the complex combination of hospital waste, chemicals, organic and inorganic materials. The expanding population and urbanization in municipal areas has led to a continuous increase in the generation of municipal solid waste. In South Africa, the most common method of waste disposal in municipal areas is landfills, with almost 90% of waste being disposed in landfills. This is due to the fact that landfilling is considered one of the simplest and cheapest methods of waste disposal. In recent times, there has been an increasing environmental concern that landfills are potentially a sink for persistent heavy metal contaminants. This is potentiated by the continuous disposal of solid waste material in landfills, which may result in heavy metal concentrations exceeding natural background levels. Soil contamination by heavy metals is further worsened by a number of landfills in South Africa operating without proper leachate collection and treatment facilities. As yet, very few published work is available on the contamination level of heavy metals in landfills around Limpopo, particularly the Weltervreden landfill site. Thus, there is a need to evaluate the concentration of heavy metals in the soil. The study answered the following question: what is the concentration of heavy metals in the soil at the Weltervreden landfill site, with the objective to assess the contamination of soil by heavy metals? Our study site is located at the Weltervreden landfill site in Limpopo province, South Africa. The study site is approximately 29 hectares in size and has been in operation since 1998. It is further characterized by semi-arid climate with a mean annual precipitation of 478 millimeters and a mean annual temperature of 28.1 to 36.8 degrees Celsius. During collection of cell samples, 2 areas were distinguished within the study area, namely the landfill site and an area located 100 meters from the landfill site, referred to as the reference site. The reference site served to compare heavy metal concentration to the landfill site. The soil samples were collected at a depth of 0 to 30 centimeters using grid sampling. The soil samples were then air dried and passed through a 2 millimeter sieve before being digested using concentrated nitric acid and hydrogen peroxide for ICP analysis. To determine the extent of heavy metal contamination, the contamination factor, which is the ratio between the metal content in the soil sample to the background value of the metal, was determined. Using the contamination factor, the pollution load index was determined and the ecological risk factor together with the potential ecological risk index. Visual distribution of the heavy metals in the landfill site and reference site was explored using box plots. A t-test was used to compare the means of the soil heavy metal data determined from the reference site and the landfill site. The significance level was set at p less than 0.05, and all statistical analysis was performed using GeneStat 21st edition. The results show that the concentration of arsenic, cadmium, chromium, and lead were significantly higher in the landfill site compared to the reference site. However, the arsenic and cadmium are the only heavy metals which were found to be above the permissible limits, which the limit is represented by the red line, with chromium and lead being within the limits. The high levels of arsenic and cadmium is a very great concern, since these heavy metals are considered to be toxic even at low levels. The source of the high levels of arsenic and cadmium can be attributed to the improper disposal of waste containing high quantities of arsenic and cadmium. Figure 4 shows the concentration of cobalt, manganese, and nickel, which were found to also be significantly higher, with the concentration to be significantly different, with the concentration in the landfill site being higher than in the reference site. These increases can be attributed to the leachate migration at the landfill site. The concentration of copper and zinc were also found to be higher in the landfill site, with iron showing a decrease in the landfill site compared to the reference site. Although these results were found to be not statistically significant, the high levels of iron in the reference site can be due to the geology of the area, since the landfill site is characterized by biotite, which is a mineral known to release subsequent amounts of iron during weathering. For heavy metal contamination assessment, Table 1 shows mean contamination factor, with the contamination factor of iron being 0.64, which is less than 1, indicating pollution, while the other heavy metals had a contamination factor of between 1 and 3. The low contamination factor of iron can be due to the fact that iron, unlike the other heavy metals, iron biodegrades in the soil. Figure 6 shows the ecological risk. the heavy metals, the individual heavy metals from cadmium to cobalt shows the ecological risk factor at the landfill site, while RI shows the overall potential ecological risk index for all the heavy metals. The ecological risk factor of cadmium was found to be 60, which indicates moderate ecological risk factor. The potential ecological risk index was found to be less than 150, indicating that the landfill site has a low potential ecological risk. In conclusion, the disposal of municipal solid waste at landfills has a considerable effect on the concentration of heavy metals in the soil. Arsenic and cadmium in the landfill site exceeded the permissible limits, indicating contamination. Similarly, the contamination indexes indicated a moderate degree of contamination, which suggests anthropogenic origins and confirmed the effect of waste disposal within the landfill site. Due to the moderate individual risk associated with cadmium, it is necessary to control its discharge into the environment to reduce the ecological risk. To reduce the quantity of heavy metals being disposed at the landfill site, the municipality should consider sorting the waste materials so that waste such as metallic waste materials can be reduced. I would like to acknowledge my supervisors, the University of Limpopo and the National Research Foundation. Thank you.
Thanks a lot for that nice presentation. You took just a bit of my time with 30 seconds there, so it's 10 minutes, 30 seconds. But I'll be generous with that. So we still have 4 minutes, 30 seconds for questions, people.
Okay, thank you very much for your presentation. I think your recommendation, you said that the municipality should look for the waste material and do something with it so that it will not affect the community around that area. Are you saying that the municipality is not doing that at the moment? Thank you.
No, what I'm suggesting is that they should consider sorting the waste, because so far the only sorting I saw at the landfill site is separation of the waste material, separation of the plastic materials from the landfill, leaving some other materials. So they should consider also sorting that material so that. Okay, thank you.
Thank you, Chair. You mentioned that the disposal method mainly used is landfill. Could you please highlight some other disposal methods? Could I just put my questions, all of them, so that you answer them once?
Okay.
The second one is that the type of the metals observed in your study and the soil type at the landfill site, do you think this contamination that you found is related to the soil type? I don't know if you have went further on your studies on that. And what should be done to avoid soil contamination by landfills? And the last one is that...
Okay, because my rules were clear, one question per person, follow-ups only if there's no more people who want questions. I think she'll just take your first question and then we'll go that side.
Okay, thank you.
We might not come back to your second question because time.
Okay, it's fine.
Okay, another method of waste disposal is recycling. And then also we have incineration, which involves the burning of the waste material.
Thank you very much for an interesting presentation. Mine is not a question, it's actually a suggestion. These results are very important. So I would urge you and your supervisor to continue with the study, maybe into a PhD, and consider investigating on the same site, utilization of production of biogas using landfill sites and how that can answer the questions that you ask now, that is contamination.
Yes.
On the same site. So it's a before and after
Okay.
into a PhD.
Thank you for the suggestion.
Can we get a round of hands for Ms. Manyike? Thank you, Ms. Manyike. I think your presentation was very engaging. People are worried about waste. Okay, rushing into possibly our last presenter for now. Ms. Masete, please come up. If you are around, please be quick.
Good day, everyone. I am Maseto Hotazo, under the supervision of Dr. T.J. Mukwabane and Dr. F.V. Ramukiti. I will be presenting under the topic, the semen characterization of Bapedi sheep, a unique genetic resource. Bapedi sheep, which are predominantly found in Hasukukune, are known for their hardy body, small, and their long legs and their fat tails. However, the breed is at risk of extinction like most of the indigenous breeds throughout the world. So far, 188 sheep breeds in the world are extinct, and conservation of indigenous genetic resources can help slow down the loss of diversity and reduce extinction of the breed. Conservation of a Bapedi sheep breed will not only prevent extinction, but can be used as a model towards improving the breed. In the quest for breed improvement and conservation, it is essential to have a comprehensive knowledge of the characteristics of the breed, making characterization one of the first essential steps in conservation. The semen characterization of the breed has received little attention in South Africa. And not to say that ewes are not important, but the semen of the ram is more important since one ram can breed 35 to 50 ewes. Hence, the production of high-quality spermatozoa by genetically superior rams is important for the improvement of the overall flock. The Bapedi sheep, like most indigenous breeds, are very unique as they are adapted to the local harsh environment, nutritional fluctuations, and are resistant to disease and parasites. Such adapted breeds can provide genes needed for improving the performance and health of commercial breeds, and therefore, such breeds are the breeds of the future and should be promoted. Preliminary work has been done by Maqash, the indicated author, whereby Bapedi rams had higher motility rate than other South African indigenous rams. However, the information provided is limited and not much is known about the breed's semen characteristics and subsequent reproductive performance. The aim of the study was to characterize the semen of Bapedi ram for conservation and breed improvement, while the objective of the study was to evaluate the semen characteristics of the Bapedi ram. The hypotheses of the study are in line with the objective. 35 Bapedi rams from the indicated research stations were selected using the total population sampling technique on condition that they are above 1 year of age. The data was collected during the natural breeding season, March to May, and the data was collected early in the morning to allow the sheep to graze afterwards. The sheep were given water ad libitum. They were also vaccinated against possible diseases. The feed were grazing on natural pasture and supplemented with lucerne in winter. Semen was collected using an electroejaculator into calibrated collecting tubes and then transferred into 15 ml falcon tubes. And then they were stored into thermoflask at 37 degrees Celsius and then transferred into the lab within 1 hour of collection. The semen volume and color were then observed visually from the falcon tube, and the semen pH was then analyzed using the HANA pH meter. The semen concentration was then also analyzed using the spectrophotometer. The sperm cell motility was then analyzed using the CASA. Viability, morphology, abnormalities, and membrane integrity were analyzed using the fluorescent microscope. Data was analyzed using SAS version 9.4, and descriptive statistics was used whereby observed variables were expressed as percentages and frequencies, and measured variables were expressed as means and standard errors. Table 1 below shows the macroscopic semen evaluation of the wabedirams, and in the study, semen was characterized as cloudy to creamy with a neutral pH of 7, which falls within the acceptable range, and the volume recorded in the study was 0.9, which is also acceptable since rams usually give a volume of 0.5 to 0.7 in younger rams and from 0.5 to 2 ml in older rams. However, the concentration recorded in the study was a bit low since semen concentration usually ranges from 1.21, but in this study, the semen concentration recorded was 0.74. And it may have been attributed to the clear and cloudy semen attributed in the study since semen color is highly correlated to semen concentration. The table 2 below shows the microscopic semen evaluation of the wabedirams, and in terms of motility, only the progressive motile sperm cells and rapid moving sperm cells will be able to reach and fertilize the oocyte under natural conditions. And in this study, the progressive motile sperm cells and rapid moving sperm cells recorded were low than other previous studies. And however, the VSL and the VAP recorded in the study did not differ much, which shows that the sperm cell motion trajectory was very linear and regular. However, the higher VCL recorded in the study shows the sperm cell swimming speed recorded on the CASA. And the study recorded a higher vitality, viability, and normal sperm cells, indicating that the sperm cells recorded in the study are very fertile. And the tertiary abnormalities were the highest of them all recorded in terms of abnormalities. Table 3 below shows the definition of some of the mentioned VAPs and VSL. Results of this study concur with the indicated author, whereby Zulu rams were also found to perform better in terms of semen quality in autumn. This study has shown better vitality, viability, and morphology than other South African indigenous rams of different breeds, including Bapedi sheep, recorded by Munyai. This study has proven that age is very critical when doing the selection of breeding rams, as the use of Bapedi rams aged 1 to 7 years resulted in slightly lower motility and concentration of semen. However, the study recorded an acceptable semen volume, good vitality, viability, and morphology of the sperm cells, indicating that the method used in the study are acceptable. Mineral deficiencies caused by grazing on natural pastures and the use of an electroejaculator may have led to the slightly higher pH, since other studies have recorded semen of from 6.3 to 7.3, and in this study, the semen was neutral, not slightly acidic. And in the study, a limited number of RAMs were available. Therefore, for future studies, an increase in number is recommended. It is also recommended to use RAMs of above 3 years to achieve the best results. The results of this study, again, can be used as a benchmark for further studies and a database for developing an effective conservation improvement and selection program. My deepest gratitude to my supervisory team, Dr. T.J. Mgwabane, Dr. F.B. Ramakiti, and Prof. T.L. Diasi, and my fellow student, and the funding provision of infrastructure and funding provision and infrastructure from the indicated institutions. These are some of the references used in the study. Thank you.
Thank you, Ms. Masete, for that interesting presentation. I think the camera loves you. We can all agree on that. Yeah, if I was you, I was going to start posing and forget that I'm having a presentation. Okay, you took about 8 minutes, 20 seconds. That does give us some leeway. Can we start with a question? I can see there's a question there. Any other hands, please raise them. Okay, there's no other hands. I'll see if I can squeeze another question.
Okay, you mentioned earlier that you feel that REMs are far more important than EWs. So my question is that, are your reasons solely based on the number of females that it can breed, or there are other reasons that you used to support that?
Okay. And not to say that EWs are not important. However, in terms of conservation nowadays, they usually focus more on the semen, which is stored and freezed for later use, just in case the breed become extinct and such stuff. And also the EWs are important. In some cases, the eggs are frozen also, but this study was focusing only on the semen of the Wapedi sheep, to answer your question.
Okay, seeing that we still have a bit of time and you are the last presenter before lunch, you say that in your recommendations you need more Bapedi RAMs. Does this mean that your results that you have are less conclusive as a result?
Not that they are less conclusive, but as you notice, the sample used in the study was a bit low due to the number of the Wapedi RAMs available. Since they are at risk of extinction, we could only find 35 Wapedi RAMs, which shows that this is a serious concern, and the number should be increased for future studies.
And how would you propose that people then, going on forward, they can basically increase the numbers?
I should say that the importance of the Wapedi sheep should be characterized and published, just so people know their importance and they should breed them more. And in this case, their numbers will increase. And as you can see, they have too much potential, therefore they can be used for breeding purposes.
Okay, no. Thanks a lot, Ms. Masete. Can we please get a round of hands? Good people, it is now good times. I think we are in for a feast. It's tea time. But I've got one request if you're going for tea time. If you're a poster person, you pass by me before tea time, right? Yes. Oh, Prof. Masu. I've heard from a senior authority that, you know, we could maybe push a bit more. I'm going to ask that maybe we get one more person. We had issues of time, which is why we are asking for this. So, other senior people agree. So, then in that case, in terms of time, I'm going to ask for the next person to come through. That next person is going to be Ms. Mashamaite. Please come entertain us. Okay, I've been reliably told that we are still a bit full, so maybe if we can try and push it, if possible, to 3 presenters. So I'm going to ask that the next 2 presenters also prepare themselves. Those will be Mr. Zwane and Ms. Chapele. So just, be on standby.
Greetings, ladies and gentlemen. My name is Puleng Mashamaite, and it is an honor to be presenting a paper on mating strategies, incidents of stillbirth, and other factors affecting cell productivity of landrace and large white crosses. The paper is supervised by Dr. Z.M. Zleje as well as Prof. Otada. The structure of the presentation is outlined as follows. We're going to go through the introduction, materials and method, results and discussion, conclusion and recommendation, as well as the references cited in the study, and lastly, the acknowledgements. Okay, but before that, I would love to give a little background for mating strategies as well as incidences of stillbirth. The mating strategies are mainly focused towards improving cell productivity with a strong emphasis on selective breeding, which, however, put a constraint in breeders having to use breeding objectives as well as inadequate pubrid lines, which will lead us to stillbirth incidences, whereby we're going to have to look at several cell factors, piglet, as well as environmental factors that are simultaneously causing stillbirth incidences, whereby the intended results have not yet been accomplished, which has been limiting the other factors associated in stillbirth incidences. As we move on, we're still on the introduction, widening the breeding objectives in order to incorporate the animal welfare as very critical in selective strategies. Thus, the implementation of crossbreeding programs or schemes will require the use of adequate pre-breed lines. Also, the genetic improvement of piglet survival is a target that could have a significant impact on productivity. Hence, identifying the risk factors associated with stillbirth could help optimize reproductive efficiency. Moving on, the study aimed to evaluate the influence of mating strategies, stillbirth incidences, as well as other factors on cell productivity traits in a crossbreeding program, which it was also used to determine the effects of mating strategies on cell productivity traits, which are litter size, number born alive, birth weight, winning weight, pre-winning litter mortality, and lastly, the average daily weight. And the last objective was to determine the effects of stillbirth incidences, sire line, sire breed, piglet sex, dam breed, filial generation, birth season, as well as birth year on cell productivity traits. Moving on, the secondary data used that was obtained from a commercial pig producer in Limpopo province, South Africa, whereby the following illustrated. The first one as the landrace pig breed, and the last figure pictured below is the large white pig breed, which were used as the experimental unit in the study, following a purposeful method. The pigs were raised in a hygienic and pig-friendly environmental. Some were inseminated, and culling was mainly based on reproductive efficiency. And lastly, the pig productive husbandry practices were followed thoroughly. The table below shows the mating strategy as a treatment factor. We have 24 mating strategy, which I'm going to show in the next slides. Factors evaluated were divided into environmental as well as animal factors, and we also have a table which shows the variables that were measured. This is the illustration of the mating strategies. And this one is an illustration of variables which were determined using methods of measurement or formulas. For any that had formulas, for example, we have a birth weight as well as weaning weight, which were measured using a looking digital display. For data analysis, the Minitab 18.1 2017 software was used. A general linear model was also used in calculating the least square means, as well as a take is honest significance different procedure was also used for mean separation at 5% significance level. The table here shows the fixed and random factor which were used for the analysis in computing the models. And on the results, we have effect of mating strategies. Highlighted in yellow, we have the large white double back rows, which improved across all traits. And then we have the pre-weaning mortality highlighted in green. The ones that are highlighted in green showed a higher pre-weaning mortality. In this case, by system, the type of breed, and also the type, which breed is used as a sire or which one is used as a dam. Continuing the effects of stillbirth on sow productivity traits, highlighted in green is the number of piglets born alive, whereby as the number of stillbirth increases, also the number of born alive reduces. Then we have the pre-weaning mortality highlighted in yellow, which is positively correlated to the stillbirth. This was also influenced by severe biological effects, which could be, in this case, adequate birth weight and cholesterol level of fat at birth. Then the next table, we have the results of filial generation, whereby in this case, the first filial generation showed reduced results, whereby in most studies you find that the first filial generation will perform higher compared to the purebreds parental generation, which in this case, we can say the type of breed used as a sire or also as a dam influence the level of heterosis attained in the study. And also the level of heterosis retained in a study shows that it's not always the case that you have to continuously breed and then the crossbreeding program that is also used also as a very important factor. And here we have the results of year on the sow productivity traits. But the most important one is the last figure, whereby the pre-weaning mortality from 2009 improved, whereby it did not increase, it reduced, which in this case, it will support the study where incidents of stillbirth are supporting the results of stillbirth incidences, whereby the pre-weaning mortality are positively correlated to incidents of stillbirth. But then we can support it by explaining by management factors as well as climatic factors that contributed to the improved results of pre-weaning mortality. In conclusion, sex and piglet sex did not affect the performance of mating strategy. Also, to represent the mating strategies, double back cross landrace improved in all traits, and therefore it is advised that preservation of purebred line should be considered. And lastly, to maximize the overall production and improve efficiency in genetic selection, it is necessary to minimize the pre-weaning litter mortality, which is also linked to increased number of stillbirth. Also, and lastly, retaining the purebreds and rotating them strategically would improve pig productivity. These are the references that we cited in the study. And I would love to give my acknowledgement to the farmer that provided with the secondary data as well as the University of Limpopo. Okay, this marks the end of my presentation. Thank you.
Thank you. You and time are like twins, like exactly on 10 minutes. 5 minutes for questions. Hands, please. Okay, people are angry. They want their tea. So I'll have to ask you a question in any case, so just so that the adjudicators can mark you on how you answer questions. Okay, so you're saying that in your recommendations that farmers can preserve these breeds. Were you not dealing with small communal farmers?
No, I was dealing with commercial farmers. Normally, they're the one who are doing crossbreedings.
Oh, okay, I think I kind of got lost there a bit. No, that's fine. Thanks a lot. Can we clap hands for Miss Mashamite?